I'm Dr. David Requena, and I'm going to talk about the, the project that I did during my PhD studies at Rockefeller University. Um, so for that, first I'm going to tell you what is fibrolamellar carcinoma. Well, this is a liver cancer that was thought to be a subtype of hepatocellular carcinoma that we all know that is one of the major cancers. And hepatocellular carcinoma usually happens in like people that drink or that have some viral infection that produce injuries in the liver. But in contrast, fibrolamellar carcinoma happens in adolescents and young adults. It's considered a pediatric cancer. And it is very rare, very, very rare. It happens in 1 in 5 million people in USA. And it is not now the incidence in the world because it's very hard to diagnose. The thing is that this cancer happens in people without history of infections or like drinking, as I told you, like cirrhosis, and the symptoms are very vague. So there is a problem in detecting this cancer, and when the detection happens, it has like often already metastasized. This is how fibrolamellar carcinoma looks like. On the left side, we have a normal liver tissue, and on the right, we have a fibrolamellar carcinoma. And as we can see, it has these fiber bands that are the reason why this cancer received its name. And the tumor cells are larger than the normal cells. However, these kind of scars are common in different things in the liver. Um, So how can we treat that? When this is localized in the liver, there are options. We can get a surgery, a resection, or we can get a liver transplantation. And when this happens, 50% of the patients reach 5-year survival. The thing is that as it is very hard to diagnose, we get patients with non-resectable cancer or already metastatic, and they have like 12 months left. It's very aggressive. So we're going to deal with a cancer that happened in kids that has no treatment, and they are going to die fast. Uh, what did we know about the genomics of fibrolamellar carcinoma? So this is good and bad at the same time. Um, it has a very stable genomic landscape with very low mutational burden. It means that it has very few mutations that are recurrent.
Sorry, the slides aren't being shared online. Let me just fix that real quick.
Which slide? Oh,
There we go.
should I repeat?
There we go.
Okay. Well, as I told you, this cancer has a very low mutational burden. It has no recurrent alterations like chromosomal alterations, translocations, duplications. So on one side, uh, this presents a good scenario to study a cancer. When we have no other factors playing a role, we can investigate a cleaner case. However, to start with that, it's like being lost because we don't have a clue of what is the cause. Um, the thing is that in 2014, my laboratory detected a single recurrent event in every patient that they tested at that time. It was constant at transcript level. It is a deletion that happens in chromosome 19, but it is not constant at genome level. But somehow you end having expression of the exon 1 of the gene DNA JB1 and the exons 2 to 10 of BRCA. And what is BRCA? It's no less than the catalytic subunit of the main kinase of the cell. And this fusion protein that they found retains the kinase activity. So we can imagine that we are dealing with a fusion protein that has the properties of protein kinase A, but with a first piece that is making it going somewhere else. So then, when I joined the lab, we investigated what triggers fibrolamellar carcinoma, and the first clue was following this fusion protein. So what we did is asking, what if we remove the genes in between, the genes that are being lost in the deletion? We tried this with CRISPR-Cas, and we also tried just adding the fusion to a model and overexpressing that. So in fact, we found that both cases reproduce the cancer. So what we concluded at that point was that this fusion is what triggers the cancer. However, when we were able to detect this cancer better, not only using the histology, but also the presence of this fusion protein, some other physicians reported some cases that look like fibrolamellar, but didn't have the fusion protein. So the obvious question is, are they also fibrolamellar or a different disease? So the first thing to do was to check what is known in the literature. So we checked the other the existing transcriptomic studies, and we found that they were small data sets with a high number of unpaired samples. So this represents a statistical problem. The batch effect, when some of these studies were combined, was not addressed properly. The tumors in some of these old studies were categorized only based on histology, and we found that in one study, even 30% of the samples were not fibrolamellar. And they were done using all algorithms like if HTC can feature counts that have problems when you have a read mapping to two annotations that are overlapping, like two genes or two transcripts, instead of newer tools like Callisto or Salmon. So this ended in getting poor agreement when we compared all of these studies. So what I did is I addressed all these points using a large data set with more per samples, a proper integrative analysis, a reliable criterion to detect FLC. And in short, what I did is I know that the tumors that look like fibrolamellar and that have the fusion, they are fibrolamellar. So I'm going to start studying those. And I use newer methods. So what I did is I collected from public repositories, and I also sequenced the largest transcriptomic collection of liver cancers to date, including multiomics. And I use state-of-the-art multiomic methods to analyze each of these omic layers and to combine them. So in short, what I did is I used one group of my data for exploration, another group for refinement, and external data sets for validation. And I found genes that have the same dysregulation, I mean going up or going down in every sample in my exploration and refining data sets. And when I compare this with the validation data sets, they all agree. What are the results of that? This is how the different data sets look when we use all the transcriptome, but when we use just the transcriptomic signature that I found, they look like that. So we have a very strong group of FLC tumors at the top left and here. And we have another group of normal samples, and we have in the middle a third group of mutant BAP1 tumors. So we ended discerning from the new FLC-like tumors which ones are fibrolamellar carcinoma and which ones are not. And with that, we determine what is the actual molecular phenotype of fibrolamellar carcinoma. So two things here. I found a transcriptomic signature that is independent of the experimental processing because each of these data sets were processing a different way using a different sequencer by different groups. And we ended getting a very strong cluster independent of that. And the second thing is that what characterizes fibrolamellar carcinoma is a activation of PKA and not the fusion that I mentioned in the beginning of my presentation. So then I got about 700 genes, but this is too much, and they might be shared with other liver tumors. So what I did is the same for the four major liver cancers: cholangiocarcinoma, hepatoblastoma, and hepatocellular carcinoma. And I found that the fibrolamellar signature makes fibrolamellar cluster very strong in one corner. And this sample that you see in cyan is one of the FLC-like samples that we determined that is also fibrolamellar. So we have that what defines fibrolamellar carcinoma is increased PKA activity happening in the liver. And we have a fibrolamellar signature. But what's in the middle and what is next? How can we put these pieces in place, like the genome, the methylome, the proteome, the phosphoproteome? How can we connect the PKA increased activity to the transcriptomic signature? So what I did is a multiomic analysis where I found that there is no strong explanation between the mutations in the genome because, as we know now, there are very few mutations, and in the methylation, I found that there are just 22 genes that are hypermethylated from the protocoherine family cluster, but I found a strong correlation between the protein and the transcription. I used the fibrolamellar signature to validate a tumor model, and we developed a mice that can get fibrolamellar carcinoma by implanting a piece of tumor subcutaneously, and this is how the samples look like when we use all the genes. However, when we use only the fibrolamellar signature, we can see here that all the samples, like the passage 1 or 3, are close to the originating tumor, that is this metastasis in green as example. And the correlation was amazingly strong between the mice model and the tumor tissue from patients. So far we have here that we found no explanation between the genome, the methylome, but we found correlation with the proteome and the phosphoproteome that I'm not showing today, but we are still missing how to connect the increased PKA activity to the transcriptomics FLC signature. So we explored the transcription factors because PKA phosphorylates. So maybe this is the path. So in fact, we found that there are some transcription factors that preferentially target genes that are in the fibrolamellar signature. And among those transcription factors, between those transcription factors and PKA, I found just a few intermediate steps. And among them, there is this gene that I want to highlight, HDAC1. And why? Because when we agnostically and in parallel, we did a screening of 5,000 drugs in the tumor model that I just mentioned, we found that the only compounds that are FDA approved that can reduce just the tumor and not the normal were HDAC inhibitors. So we were connecting the points finally. Okay, so now if we remember, we saw a picture of the tumor with some fiber bands, and the bulk tumor has like some different cells in there. So I did also a single cell spatial transcriptomics. I found 3 strong clusters corresponding to each of the tumor tissue regions. As example, in this tumor slide, if we map the genes that we found at the top of the clusters, we can identify clearly the stroma, the normal, and the tumor tissue sample. So in this way, we were able to identify the contribution of each tumor each tumor cell type in the bulk tumor tissue. So to finalize my presentation, I would like to thank my former lab, the external collaborators, and if you are interested in my research, you can just search me in Google Scholar, and if you wanna know about my current research, because I'm no longer at Rockefeller University, you can email me. So that's all. Thank you.